Muscle growth, scientifically known as hypertrophy, is not an aesthetic pursuit to the human body. It is a stress adaptation response. From an evolutionary biology perspective, maintaining muscle tissue is metabolically expensive. The body will only synthesize new contractile proteins if the current environment exerts a mechanical stress that exceeds the current capacity of the musculoskeletal system. This process is governed by a complex cascade of intracellular signaling, primarily centered around a protein kinase known as mTOR. The process begins at the cellular membrane with a phenomenon called mechanotransduction. When a muscle fiber contracts against resistance, the physical tension creates shear stress on the sarcolemma. This mechanical force is detected by mechanosensors, such as integrins and focal adhesion kinases. These sensors convert the physical mechanical signal into a chemical signal inside the cell. Without this specific mechanical threshold being met, no amount of protein consumption will trigger synthesis. The signal is binary, adapt or fail. Once the mechanical signal is received, it activates the mammalian target of rapamycin complex 1, mTOR-1. Think of mTOR as the master regulator of cell growth. It integrates inputs from three sources: mechanical stress, energy status, and nutrient availability. When activated, mTOR phosphorylates downstream targets like P70S6K, which initiates the translation of mRNA into new proteins. Effectively, mTOR is the foreman that reads the blueprints for new tissue. If mTOR is inhibited due to energy restriction or lack of specific amino acids, synthesis halts completely. While all essential amino acids are required for building tissue, one acts as the primary trigger, leucine. Data indicates that there is a leucine threshold, a specific concentration of intracellular leucine required to activate mTOR. Consuming protein below this threshold may provide raw materials, but it fails to flip the on switch for synthesis. This explains why protein quality is statistically more significant than total protein quantity. The presence of insulin also plays a permissive role here, suppressing protein breakdown to allow net positive balance. For long-term growth, the muscle fiber faces a logistical problem, the myonuclear domain theory. A single nucleus can only manage a specific volume of cytoplasm. As a muscle cell grows larger, its existing nuclei cannot support the expanded volume. This is where satellite cells come into play. Mechanical trauma activates these satellite cells, causing them to proliferate and fuse with the existing muscle fiber, donating their nuclei. This addition of new DNA allows the muscle cell to ramp up protein production and support a larger size permanently. Finally, we must address the muscle full effect. After a bout of synthesis is triggered, the machinery enters a refractory period where it becomes temporarily insensitive to further nutrient intake. Data suggests that muscle protein synthesis peaks around 3 hours post-stimulus feeding and then baseline returns. This indicates that synthesis is cyclical, not continuous. Muscle synthesis is a coordination of mechanical stress, hormonal signaling, and genetic recruitment. It is a highly regulated, energy-dependent process that strictly follows the laws of thermodynamics and cellular biology.